Humanized plants?
Yes.
How do you...
I don't know. I can read about that later. Let's make a podcast episode.
Okay.
I would like to introduce
FAU-GEM.
Welcome to another episode of FAU-GEM. My name is Laura. I will be your host today, and I'm here together with
Marina.
And Maria.
Today we will present you another iGEM project, as we usually do. For our new listeners out there, remember that our usual outline is first talk a bit about the team, then about the problem that they address, then we focus on their projects and maybe add some collaborations that they had or some interesting facts in the end. So if you're okay with it, maybe you can present the team, Marina.
Yes, so today we're talking about a team that is from the University of Leiden, and their project is S.P.L.A.S.H. So it's in 2019 they were participating in the iGEM project, and they won the Overgrad Best Manufacturing Project. They were 11 students from various disciplines like biology, life science and technology, molecular science and technology, but they were also like mixed bachelor students and master students, and in general they had 7 advisors.
Which I think it's like a super big name, right? I mean,
Yes.
7 advisors.
7 advisors for 11 students.
Yeah. Amazing.
Yes.
Okay, so now we will move on to explaining the problem that they addressed. And to do it, I have a riddle for you girls. I'm gonna ask, let's see if you know something that it's pretty common. I think it has happened to almost everyone, not to say to everyone, you know, scientific way of thinking. There's always exceptions. And it's something that it's pretty common, especially when you're young and, you know, you don't know how things work. And it could happen a lot of times in the kitchen. Do you know what could be a problem? It can be fixed, it depends on the degree, but sometimes it could be fixed quite quickly, other times not so quickly, but what could it be?
Since you're saying degree, I'm thinking burn wounds, maybe?
Yes, exactly. I'm talking about burn wounds, and the problem that the team of the University of Leiden addressed was severe burn wounds. They actually have like data here, fun fact. There are the cause of around 300,000 deaths annually. I think it's quite a high number.
Yeah, that's a lot.
Really high.
And actually, out of all these deaths, around 42 to 65%, it is usually because of bacterial infections or posterior infections.
Just a question, maybe it's of importance for our audience. What are considered severe burn wounds? So what percentage of the skin has to be affected for it to be a severe burn wound?
I think there are like 4 different degrees of burns. Like the first degree is like probably like the rather easy one when you put your hand on the stuff, or I don't know, burn your hand in the oven, like nothing too severe. And then like 2nd, 3rd and 4th. I think 4th degree is like the ultimate pain, when it also affects like maybe your muscles and your bones already, like there's like the worst case. So I think maybe severe wounds starting at 2nd or 3rd degree.
So it depends more on the severity of the symptoms more than on the extension,
so
right?
I think like for the 1st degree there are like almost no blisters, and the more blistery, the more red it gets, the more severe it is.
The worst, and then you get like the highest degree burn. But
Makes sense.
good question, Maria. Okay, great. So this was kind of this is the basis of the problem. And the thing about burns is that, well, I guess you can imagine, and with the kind of description that you gave, Marina, like if you have even the bones affected, I think the victims or the people that suffer this kind of burns obviously suffer not only physical but also like psychological consequences, and probably they even sometimes have to face disability of some kind. They completely lose function of I don't know, an extremity or something like that,
like hands or legs.
Exactly.
And
So-
maybe they even have like problems concerning their work. Maybe they can't do the work anymore that they did before, or maybe they're too long in the hospital and need too long for a recovery time that they get problems economically.
Yeah, definitely.
And how does treatment work, or what are the problems with treatment?
That was what I was going to ask, but amazing. So usually when patients face this kind of problem, they usually sometimes go to donor skin application, or kind of like, skin transplantation. But the problem with this is that there's sometimes problems with availability, so it's kind of difficult to get this kind of skin, and the kind of requirements that you need are very, very specific, like, you have to be careful so that you don't get also like graft versus host disease because of the transplantation and so on. And besides this graft versus host disease problem, we also have another challenges, like for example fluid loss, increased risk of wounds, like you have like a completely new skin there and you can get infections, and you can then, because of this, have even further functions that the skin usually displays, like, I don't know, protection barrier functions that are sometimes also impaired. So it is kind of hard, and this is why this team focused on a new idea, and it's because the WHO, so the World Health Organization, also says that sometimes you can use substitutes of this donor skin application, and one of them are hydrogels. So do you girls know what a hydrogel is?
Yes, I think hydrogels are hydrophilic polymer chains that have the ability to store large amounts of water due to their hydrophilic nature, and that also means that they increase in volume when in contact with water.
Oh, water, yes, that makes sense because of like the Greek word hydro. So
true, very true. Everything makes sense now.
So a hydrogel can like absorb a large quantity of water and has like a good potential, therefore, as I applied in biomedical purposes. So they thereby promote the fibroblast proliferation and the keratinocyte migration that help like for the recovery.
Exactly, yeah. It's kind of what we explained earlier, like the problem that we have when we use this donor skin application is that we still have sometimes significant fluid loss, and with this we retain this kind of wound exudate that we generate or that we naturally produce to prevent infections and to cure, recover, and we kind of retain this, and this will help exactly with what you said, Marina, with fibroblast proliferation and keratinocyte migration, which are cells that are basically essential for the skin. Amazing, yeah. But there is actually a problem with hydrogels. Can you imagine what this could be?
I think that the materials needed for the hydrogels could be rather expensive and also maybe have poor mechanical properties, and that then would lead to a faster contraction of the graft.
Yeah, this is usually what happens, and so therefore there has been like a lot of research lately to try to circumvent this kind of problem, and there has been, or a lot of scientists have looked actually at proteins that we have also mentioned in another podcast episode, which are spider silk proteins.
Oh, that's so smart.
Yeah, it's very smart because actually, like, if you can imagine of the spider silk, they can support like a lot of tension. They're like really thorough, rigid structure. Well, rigid in the sense that they can support like a lot of weight. And so there's like a lot of studies going towards that goal. However, our team focused on different kind of proteins. These are actually proteins that derive from cephalopod, particularly Dosidicus gigas, sorry for the Latin pronunciation, that's the name of the species of this squid. And so let's dive into the project then. So they decided that instead of focusing on this spider silk proteins, which are called spirines, they focused on saccharins, which are this kind of proteins that are derived from this squid.
Maybe that's a good opportunity to explain what S.P.L.A.S.H stands for, because it's also in the name.
Mm-hmm, exactly.
So S.P.L.A.S.H stands for Saccharine Polymer Layer to Achieve Sustainable Health.
Exactly. So this is actually also really interesting because obviously, as I said, they have they're really similar to this spider silk, and therefore they're also biodegradable, and they have all of these characteristics that are also nowadays very important and very essential.
And how does it work now that they have like the toughness and that they form such a matrix?
Mm-hmm. So in the genome, to produce these proteins, the gene actually have like two modules, and in the first one, it actually codes for the hydrophobic amino acid alanine, and these residues, they through hydrogen bonding, they're able to form these beta sheet structures, and this is actually they even form nanocrystals, and this is what gives the property of this toughness, this rigidness to the whole protein. And in another module we have flexible chains, and here we have glycine, tyrosine and leucine amino acids, and this is actually what forms this kind of amorphous matrix, so it's actually where these nanocrystals are actually embedded. Okay? Makes sense. I actually think it's like really smart, and the fact that nature is able to produce these things,
that's enormous. Yes,
yeah.
What nature does, wow.
Mm-hmm. And actually also a really interesting thing is that these saccharine proteins are actually able to self-assemble. So they are actually like the spider silk one. This is why they're like really similar.
So they are synthesized and then they just assemble in the right way without any external help.
Exactly. Okay. That is what self-assemble means. Yeah.
Do you know what those proteins originally do in the squid?
So in the tentacles you have these rings, and they are actually part of these rings, and these rings are called suckers, and they do what the name actually implies.
They suck.
Exactly. So they are basically used to latch and take down like a prey, and that's why we have this kind of, they form these teeth, between quotes, these teeth, so that they can be rigid and actually
stick to the target.
Stick to the prey. Yeah, exactly. Very good. So going back to the project, there's actually like one question you haven't asked me, because I said that they're like really similar to the spider silk proteins.
Oh yes, why don't they just use spider silk proteins then?
If it's so similar. Exactly. This is actually a really good question, because as I said, like there's much more research focused on these spider silk proteins, but spider silk proteins have a bit of a downside, and is that they have like a really high molecular weight, between 300 and 450 kilodaltons, and this actually has some problems when you want to produce these proteins then in vitro. And saccharines actually have a lower molecular weight, between 5 and 57 kilodaltons.
Oh yeah, that makes the difference.
Exactly, and this is what actually makes the production process much more efficient.
So they can be produced, for example, in E. coli or something like that.
Exactly, yeah. And this is actually the project. So they actually decided to produce these saccharines on E. coli. Here, they actually asked themselves a question that I also asked myself while reading their wiki, which was, if this is actually a protein that is produced in eukaryotes, why do you use like a prokaryote system, right? Like, why would you choose E. coli? Because the protein production system, it's not the same in prokaryotes and eukaryotes, right?
Yes, and for example, there are post-translational modifications that occur in eukaryotes that can be done in prokaryotes. Did they say how they circumvented that problem, or was that even a problem at all?
Yeah. So actually, this happened like, first of all, they talked with different research groups, like, also kind of human practices part of the project, and they talked with one research group which was able to produce saccharin 19 in E. coli. And so they actually decided to use E. coli as a model organism to produce saccharin. However, they also received input from other research groups where they were also told that maybe they should also do it in other organisms. So they used S. lividans as a gram-positive prokaryote to produce it, and they also had to think about what you mentioned, Maria, post-translational modifications. So they actually ended up also doing it in Saccharomyces cerevisiae, which we said is a yeast. So after all of this, they were then able to produce and purify saccharins. They were able to do it for saccharins 19, 12, 8 and 9 mainly. And after this protein production and purification, they did this all by using an inducible promoter, quite important, and they were able then to produce everything with bioreactors to produce like great scale, or to manage to get like great scale production of all of this.
And for the hydrogel, then they used a mixture of like the proteins.
Exactly,
like different sacharins.
The different sacharins, yeah, exactly. And so this all ended up in this way, and they actually also talked with different companies to see if, and then this could be like a commercial option, and to see if this could get into the market. So they were quite successful, I would say. And with that, we jump to our last section, which is more kind of like outreach, collaborations, and so on. As we mentioned, they talked a lot with different research groups, which obviously always give interesting feedback, like the thing with the post-translational modifications, like, hey, have you thought about that? That is quite important. But other than that, they, as we mentioned throughout the whole podcast, used protein really similar to spider silk. So you can see that spider silk and similars are quite common in iGEM competitions, as we also mentioned in our second episode. So if you haven't listened to that one, you should definitely go and check it out. And and they also were able to produce these biobricks with these plasmids with promotable, inducible promoters, which are mentioned as biobricks. And with that, this is the end of our 6th episode. Thank you so much for listening.
Yes, thank you for listening.
Bye. Stay curious and stay tuned for the next episode.